Hello all and welcome to the Jubilant Biosys webinar on animal models of NASH, challenges and way forward. We will be beginning the webinar in just about 2 minutes, and while we wait for other participants to join in, we will post a survey question on your screen. Please make time to chip in with your responses. Thank you. We'll begin with the webinar with the next slide and talk about the house rules for the day. So we'll be recording this session, and Jubilant Biosys holds all right for promotion and distribution of this recording. For ease of execution, all attendees' microphone and cameras will be disabled throughout the webinar. If any one of you would want to post any questions to our panelists, please use the Q&A section. This will allow for an uninterrupted experience for everyone watching this webinar. Time permitting, towards the end of this webinar, we'll have a Q&A section, and if we could not answer your question during that time, we'll make sure that someone from our team gets in touch with you with our responses. So let's get started. This is me, Saurabh Kapure. Brief bio. I'm a pharmacist and a management graduate, and I have been selling India to the world for the last 19 years. I have worked across global markets and part of the Jubilant Biosys ecosystem for over 6 years. I am also on the board for multiple Series A biotechs working in the space of inflammation and NASH as a Jubilant Biosys representative. Talking about the session, we'll have a brief video about our newly launched chemistry research campus for you to view, followed by which we'll talk about our integrated drug discovery capabilities at Jubilant for again a very brief time. After that, I'll hand over the baton to our speaker of the day with a brief introduction. He'll take you all through the webinar that revolves around animal models of NASH, challenges, and way forward. Towards the end, we'll have another interactive poll and then close it with a Q&A session at the last. Talking about the Chemistry Innovation Research Center, Jubilant has launched this 500 FTE chemistry site earlier this year, and this site comes with several unique features which are first in the industry. So we have digital fume hoods which are enabled with webcams, temperature sensors, as well as proximity sensors, all of which allow for better monitoring as well as safe execution of the work. We have analytical and purification support system which has been decentralized and placed at each floor, allowing for operational efficiency. We are a highly digitized infrastructure in this site where we use ELN, e-analytics, and several digital tools for inventory and building management setup. Keeping in mind the COVID-19 pandemic, we have also invested in advanced air filtration and viral purification systems. Also, we have automated and very safe pipe solvent dispensing, again decentralized from the central solvent store to each floor, allowing for better operational efficiency. Last but not the least, we also have a pneumatic sample distribution system across the site for NMR samples to be deposited in the central NMR warehouse. This again takes care of ease in material flow and avoids people movement across the site and helps us keep up the productivity. So this is a brief summary of our newly launched Chemistry Innovation Center. And with that, I'll actually take you through a brief video talking about these features. So the short video will begin now.
Welcome to Jubilant Biosys, our integrated drug discovery center here in Bengaluru. People often ask us, How do we achieve successful integrated drug discovery at Jubilant Biosys? How have we been able to consistently deliver well over 80 integrated drug discovery projects and many clinical candidates over the years? Our work here is multidisciplinary and demands attention to detail, dedication, and diligence to deliver discovery success.
Our 4D philosophy, time management with high quality data, drives us at Biosys to offer IDD with speed and efficiency.
I will briefly run through the different processes that gives our IDD flow from target validation to lead candidate selection. We realize your ideas for a drug starting from a concept and then shaping it up into a hit, lead, and ultimately a preclinical candidate.
Computational chemistry analysis and structural biology, including protein science and crystallography, to support the design of molecules.
ADME and pharmacokinetic studies to discover more about the molecule, with in vitro and in vivo pharmacology also coming into play. Use of a rapid-fire throughput screening system gets results faster. RB-Trap to swiftly identify metabolites. Lead optimization and design enhancement to accelerate optimization of the molecule and quickly realize a clinical candidate.
Medicinal chemistry works hand in hand with DMPK to speed up design.
Structure-based drug design with target engagement and disease models helps us to understand the molecule better.
Efficacy and tox studies, including AIMS and HERC, play their part in further optimizing the molecule to obtain the ideal candidate. Testing our compounds exhaustively is the key to successful IDD.
Successful IDD requires a team. Our team uses its knowledge and procedures established at Biosys to implement the process, which puts quality and our 4D promise at the center of our working process. We have disease biology and drug hunting expertise in oncology, metabolic disorders, CNS, and pain and inflammation. Our team approach to working collaboratively with customers makes us successful at IDD. Please contact us today and let's talk IDD. Thank you.
Thanks for bearing with us on this video. With that, I'll make a quick introduction to our speaker, Dr. Sanjay Kadnur. Dr. Kadnur is a drug hunter and has experience of over 16 years working in the metabolic and the inflammatory disease space, in vivo pharmacology being his forte. He has worked in several Indian pharma as well as biotech companies before joining Jubilant a few years back. Academically, he is a MPharm pharmacology, and he has also pursued his PhD from LM College of Pharmacy. At Biosys, Dr. Kadnur has led several integrated programs spanning the inflammatory disease and metabolic disease space and taken them to phase 1 or phase 2 studies in the current scheme of things. He also has contributed towards several publications and patents to his credit. So with that, over to you, Dr. Sanjay Kadnur.
Thank you, Saurabh, and welcome and good morning, good afternoon, good evening from wherever you are taking the call. Today we'll talk about animal models of NASH, challenges and way forward. Moving to the next slide. Talking about NASH, that is a non-alcoholic steatohepatitis. This is a kind of hepatitis which is different from alcoholic hepatitis, where people are affected with steatosis and later with the fibrosis of various stages without any influence of alcohol, meaning that these people are suffering from this steatosis and increased fat deposition and fibrosis over a period of time with diet and sedentary lifestyle and stress and environmental influences along with genetic influences. So what you see in this particular slide is, if you see in the bottom left panel, the global map of people affected with NASH are clearly recorded here wherever the data is available. If you see, irrespective of the region of the world, depending upon the people, there are various degrees of NASH which are affected, either 20% or more in several countries, both in advanced and underdeveloping or developing countries. This indicates that it is a global problem. And if you look into this particular disease, as such, NASH is a severe disease where the normal liver, over a period of time, when the fat accumulation takes place, it turns into something called as non-alcoholic fatty liver, or the steatosis occurs here. And over a period of time, if people don't take care, adequate attention on the lifestyle changes, they lead to something called as NASH without fibrosis, and later they again move on to progress to something called as NASH fibrosis, where you have the stages like F1, F2, F3, and again, if that is again not taken care, which is again moved to something called as NASH with fibrosis with cirrhosis, and later leads to hepatocellular carcinoma. If you look into the last two things, that is the NASH with cirrhosis or the hepatocellular carcinoma, these are the patients in recent times, people are requiring the liver replacements, which is adding to the burden which is already piled up. So what it tells is this is a condition of metabolic diseases which is affected in patient population who are already obese or hyperlipidemic, diabetic or cardiovascular conditions. So these are some of the comorbidities which are associated with NASH. And if you see this progression, it is, as I said, it is a progressive disease. So where you are seeing from NAFL to various stages of fibrosis, it is a slowly progressing or fastly progressing. It depends on various conditions. So there are fast progressors or slow progressors. If you look into the global population of people who are affected with NASH, it clearly tells there are several million people, maybe close to 100 million plus people are available in the global today who are affected with NASH. This brings to social, health, and financial challenges to the global community to handle this particular disease. So what is required in this particular condition to treat the drug is given by various drug administration agencies. They look for a drug which is highly beneficial with minimum risk, because this should be treated for several years or several decades to have the conditions to be maintained or reversed. So in this case, what is expected from the regulatory agencies is resolution of steatohepatitis and then reversal or regression of fibrosis, which is there. Moving to the next slide. With this challenge in the drug discovery and development, so people have taken several molecules into the clinical trials. If you see in this particular slide, what you clearly see is that there are several drugs which have moved into the various stages of clinical trials. What we show here is the phase 2 and phase 3 clinical trials, where you see the red dots which are representing the molecules which have been advanced either at phase 2 and phase 3 or registrational trials and they have failed. And some molecules or many molecules which are lined up and all are in active testing. So if you see the particular slide here, what you see here is there are several mechanisms people have targeted, which are targeting the metabolic diseases, the inflammation or the fibrosis or the combination of that. If you, there are several modalities starting from small molecules, biologics and newer modalities. If you look into the combination drugs, either 1, 2 or multiple drugs are combined and seen there for their effects in the NASH. If you see, this brings to a bigger question. Having 100 plus million people who are affected with NASH, there is a high need of any drug for treatment of NASH. But with this particular crowded space, still people are finding it difficult to find a successful drug which has enough benefit ratio as compared to the risk. So this brings to a particular attention for the drug discovery community to understand and deep dive. What is understood all these days is that the molecules which have advanced in recent times or in the past, either they had a lesser efficacy or a potency, or they were not adequate enough so that they could not translate with whatever the hypothesis which was generated. Moving to the next slide. So for any drug discovery and development effort, you need two things. That is, you need to have a ideal target, which you are making out of a program out of that. There you need something called as in vitro cell-based assays or the in vitro assays, and then you also need to successfully test these molecules into animal models. So what in the NASH has offered over the years or the decades is that you have something called as in vitro cell-based assays or the biochemical assays, where you started in the late 80s, people have started with FG2 cell-based assays, and then people have taken something called as isolated primary hepatocytes. And later, people also came up with more complex systems, the human stellate cells and isolated primary human hepatocytes. And more recently, people are talking about organ on a chip or the organoid models, where you have more complex multicellular systems, which also multiple flow, which resemble the physiology of the liver and many conditions of the NASH. And if progressing the molecules from various models of in vitro testing of the NASH, so people generally move it to something called as animal models, where you have mouse models or the non-rodent models. So basically, people have tried to mimic the NASH condition in the animals by providing high-fat diets or chemical induction or genetic induction. Many people have also tried to capture these particular NASH conditions by combination of these things. So there are several progress in the animal models of NASH. Mainly the animal models of NASH are dealt by using the mouse as a model, which is very convenient to utilize. But what you see is over the last several years, people have tried to mimic some conditions of the human NASH. But with whatever the advancements people have taken with a lot of effort, still there is some gap in identifying animal model which will completely mimic what is seen in the human NASH condition. But this progress is very much advanced, and in the most recent times, people have come up with more models which are very, very interesting. Moving to the next slide. Talking about challenges in NASH animal models to replicate human NASH. So what you see in this particular slide is the histology figures of human NASH and the animal NASH, where you are seeing the fibrosis in the top left corner in the human NASH, which clearly shows various degrees of fibrosis. And if you see in the top center panel here, you also see a fibrosis in the animal model, which resemble to a certain extent to what is you see in the human NASH. In the bottom, in the middle left panel, you see the HND stains where you have the steatosis, which is again most probably you can see a better correlations both in human and animal models. So what you see in the last, that is the bottom left and the bottom center, is the inflammation marker, where the macrophages are seen in both these places, where you can see again some resemblance. So what is the challenge here is the lack of translation. What I mean by lack of translation is when the molecules are tested in the animal models of various models, whatever it is available today. So these molecules which have shown efficacy in these animal models, when they are taken into the phase 2 or phase 3 trials, they have not shown the similar kind of efficacy what is shown in the animal models. So this is a burning challenge the drug discovery industry is facing today. And duration of the studies, both in clinic or in the preclinical studies, are very lengthy in this case. There are some models which are short in nature, but most of the models, what you are having high reliability, have very lengthy duration of studies. So this brings to a very big cycle time of testing drugs and filtering the successful molecules for later stage studies. Progression of disease. As I said, NASH has various as a progressive disease. It has the fibrosis component, which runs from F1, F2, and F3 stages of fibrosis. So this is a progressive disease. The progression is either fast or slow in the human conditions. And what you see in the progression of animals also is very different. So what is important here is to maintain the same kind of progression in animal models as compared to the human NASH. Coming to the treatment paradigm in patients. So why it is very important is in the human real world situation, as NASH people are having a lot of comorbidities like diabetes, hyperlipidemia, obesity, and cardiovascular conditions. When people are treated with in the clinical trials with candidates of NASH, they're not alone treated with for NASH, but also with the comorbidities. But when you see the molecules in the earlier stages, when it is tested in the animal models, only the molecules which are treating the NASH is taken and tested. So there is a gap between what you test in the animal models and what you test in the human real world situation. And the heterogeneous patient population, as I said, it is a global problem where people from various degrees of diet or ethnicity or genetic or the climatic conditions or the environmental influences and various other stressors are there. But irrespective of them, in the global population, you see several millions of patients who are associated with this. But what you see in the animal models is that you pick very uniquely highly controlled animals, which may not exactly mimic what you see in the human population, but it will mimic some portion of the human NASH condition. So it should be very clever team to understand what is available from a particular model and what should be read out of that particular model for human clinical trial advancement. Moving to the next slide. So with these all challenges available to the drug discovery community, what are the approaches people have taken to overcome challenges in the NASH animal models is very essential to discuss now. So if you look into various categories, what people have taken as an approach, people have adopted artificial intelligence techniques. In this case, I'm just talking about NASH pathology assessments in more recent years, and multi-omics approach to identify the targets and biomarkers. And it is very essential for people to get some recapitulating clinically effective drugs in animal models. So this can be done only when we find the first drug which is efficacious in the human clinical trial, especially in the phase 3 or the registrational trials, and take it back into animal models and identify, when you test these molecules in the efficacy models, what is the different signatures you are seeing between an animal which is efficacious in both in animal model and the human NASH condition, and for those other molecules which have shown efficacy in the animal models and failed in the human clinical trials. So there will be some patterns of the signatures, which is very important for us to identify and then apply it widely into other things. So testing in few models with target of interest expressed in relevant tissues. So this is very essential. If you see in the past several decades, people have looked at various programs based on various approaches. So one of the things which happens in the drug discovery or development condition is that the targets may be of relevance, but it is of various degrees of expression is seen in these particular targets. In this case, it is mainly in the liver. So say, for example, there is a target which is very important for the human NASH, but if their expression is limited in the human animal models or the animal models, which will not exactly correlate that of the clinical human NASH. So one should pay attention to understand what degree of this protein is expressed and its relevance in the animal model, and adopting human biomarkers in animal models. So this is very essential. As I said, this is a disease which is affecting several million population across the globe. You need to have markers to stage the disease and the prognosis, and also to understand how it will be applicable for various NASH conditions. So the same kind of markers should be adopted in the animal models, so that later when these animal models results are taken into human clinical trials and later into the clinical practice, these markers can be widely adopted. These markers should be cost-effective, sensitive, specific, so that it will be useful all the way from animal models to the clinical practice in the world. So liquid biopsies are very important because, as you know, at present day, the liver histology is the gold standard both in animal models and the clinical trial applications. So this cannot be practiced in the real world situation. As you know, the technical expertise or the infrastructure or the cost associated with this and the inconvenience to the patient will not allow this to happen. So it is very essential and it is very high time to get some liquid biopsy or markers, which is essential to recapitulate what is seen in prognosis in the NASH, so that it can be taken into human clinical trials and for real world practice later. Moving to the next slide. Talking about AI in NASH pathology assessments. As we all know, artificial intelligence techniques like deep learning and machine learning have been applied in various fields within multiple industries, especially in the drug discovery and development. This is also widely applicable. If you take the specific case of NASH, what you see in this particular case is, if you see a slide here where the liver histology slides are taken and the whole slide imaging of this is done, and which will be subjected for pathological scoring. And along with that, the artificial intelligence techniques are applied, and along with the pathologists, these methods will train the artificial intelligence techniques to score very similar to what is done by the pathologist. over a period of time, the pathology scores and the AI-based scorings will become very similar. And there is also, you can remove the biasness which is seen by one or two people who score these by pathologists. And it also comes with a digital slides where you can read these slides for a later time, and also with a lot of rich information which can be used for various applications in the future. This is one of the AI applications which you can find, and in most recent times, the drug discovery industry and the regulators around the world is welcoming these important changes. Moving to the next slide. Talking about multi-omics approach in NASH. So multi-omics approach in NASH is very essential. As I said, this is a multifactorial disease, and there is a huge global population who are affected with this particular disease. It is a high time for us to understand what are the causative things which is affecting the NASH, and also pinpoint the genetics and the proteomics and the metabolic signatures associated with these. There are a lot of efforts which have gone in the recent past, which has helped the drug discovery and development industry to pinpoint certain things. So the causal genes are identified in most recent years. If you see in the right side figure, you have various targets of interest which have the causal in the nature. And it is known in the drug discovery and development industry that whenever there are causal genes which are responsible for the disease, if you intervene this particular kind of targets, the successful outcome of these particular targets or the projects around these will be very high. If you see that these kind of targets, when they are expressed in the human liver and as well as in the mouse liver, to a greater extent, then modulating these targets with different approaches will be helpful, and also getting the metabolic signatures and the other signatures using the artificial intelligence techniques will help us to get clear markers and pathway modulations, which is essential for human NASH. Moving to the next slide. Talking about adopting biomarkers both in rodents and humans. This is a very essential topic as of now and in the coming days, because as we know, there are several markers, but having so many markers, it is very essential for us to pinpoint which of the selected few markers which will be helpful both in preclinical studies, in the animal models, human clinical trials, and in the real world practice. So it is now what we have at our disposal is the histopathology and the markers of interest from both the invasive and non-invasive techniques, and also imaging techniques. As you know, a combination of these is available, which will give a better insight at this point in time. As I said earlier, sensitivity, specificity, cost effectiveness, and widely applicability with less technical expertise is the game changer in this particular field. So there are a lot of advancement which is taking place. And in most cases, a companion diagnostics is also built so that people don't need to get into liver histology, so that a lot of inconvenience and the variations associated with that can be avoided. So these are some of the things which you can see here, both the invasive, non-invasive, and the imaging techniques, there are a lot of advances in this particular case. Moving to the next slide. So talking about all these challenges and what are the way approaches people are talking, I'm just switching gears now to Jubilant Biosys NASH research capabilities. As you know, Jubilant Biosys is very well known in the field of drug discovery services, where we have successfully delivered several preclinical candidates which have advanced into clinical candidates, among which metabolic disease is one of the areas out of which the NASH drug discovery is active research at our center, where we support programs with target of interest from the client, and then we provide the appropriate in vitro assays and the animal models of NASH so that they can fit for purpose, stage appropriate, and customer-centric suggestions will be made by our drug discovery experts who will support these assays. If you look into our in vitro assays, we generally come up with target-based biochemical and cell-based assays, which is very much useful for any particular targets in the initial stages. And then this will be subjected and supported by various more complex systems like hepatocyte cell interaction models. And also we provide something like lipid inflammation and fibrosis markers, target engagement, and pathway analysis modulation models. So when the program matures into something with a good in vitro profile and adequate ADME and PK, the molecules will be advanced into animal models of NASH. Here, our technical expertise of the team with animal models of NASH will be very essential for any drug discovery program to be successful. where we have developed some of the animal models which are available for the global community, which are a part of chemical induction or the diet induction or the combination of diet and chemical induction models. So to name few, we have the NSTZ and HFD-fed NASH model in the mouse, and we also have something like HFD-induced NASH model in mouse and CDA-HFD diet-induced NASH model in mouse. So these are some of the models what we have at our disposal. Moving to the next slide. So now switching gears to one of the models, that is neonatal streptozocin-treated high-fat diet-fed NASH model. So this is a model which represents the chemical induction and the high-fat fed or the dietary modifications in the model. So in this particular model, if you look into the study design, what we have is these animals, when they are 2 weeks of age, they'll be taken and administered with streptozocin. And at about 4 weeks, these animals will be put onto high-fat. And later at 6 weeks, these animals will be randomized based on the parameters and then will be subjected for treatment for next several weeks. At the end of the study, we sacrifice these animals and collect the liver and the plasma and various other things for the biomarkers and various analysis. Moving to the next slide. So in this particular model, talking about the liver health, as you know, NASH is very specifically associated with the degradation or the changes in the liver health. So what you see in this case, what we have done in this case is that telmisartan is the test drug which we have taken. So in this case, if you see the yellow bars, they clearly represent the disease animals where there are clearly elevation in the plasma ALT, AST, or the liver TG/TC, which clearly tells that, or relative liver weight, which clearly tells that the liver conditions are abnormal in these animal models. And when these animals are treated with the test drug, in this case telmisartan, you can see there is some reduction in some of the parameters, indicating that this telmisartan has beneficial properties in this particular model. Moving to the next slide. So talking about the histopathological assessment in this particular model. So what we showcase here is the hallmarks of NASH, that is the steatosis, inflammation, and the hepatocyte ballooning. So which are very key things in this case. And as you see in the top panel, we have the HND staining, and then in the bottom panel, we have the serous red staining. In the top center panel, you can see the disease animals where you have the clear indications of these animals are subjected for showing the steatosis and the inflammation and the ballooning. And if you see the top right corner where you have the animals treated with the test drug, where you can clearly see there is a significant improvement seen with steatosis, inflammation, and the ballooning, what you see in the diseased animals. In the bottom panel, we also showcase the serous red staining, which talks about the fibrosis. Here also you see some improvements with the test drug treatment. Moving to the next slide. So talking about the analysis of gene markers in liver samples, as you know, fibrosis, inflammation are very key markers which are altered in the disease models, and in the same thing you can also see in the human NASH. So what we showcase here is some of the markers of the fibrosis and the inflammation. Again, what you see here is the yellow bars, which represent the disease control, which clearly show there is elevation in these markers. And when treated with the test drug, in this case, some of the markers show significant reduction, indicating that this particular reference drug works through these mechanisms. Moving to the next slide. Now, switching gears to HFD diet-induced mouse model of NASH. This is a purely a dietary model where there is high fat. You know, in the globally now, most of the population in the recent decades have adopted to high fat diets or the Western diet, where because of which maybe possibly there is a lot of people who are getting into NASH or the metabolic conditions. So to mimic that, what we have done is we have put these animals into high-fat diet for about 32 weeks. This is a very long time. And then at this point in time, you randomize these animals based on body weight and the ALT and the AST. And this will be animals will be treated for the next several weeks with a test drug. In this case, we have used bempedoic acid. And at the end of the study, we sacrifice these animals, collect the liver, and various other blood and other parameters will be looked for modulation of the test drug. Move to the next slide. Talking about liver health, again, what you see here is there are several parameters which are mentioned in the diseased animals where it is in the yellow bars. So these liver health parameters, they're all deranged in the diseased animals. And if you see when these animals are subjected for bempedoic acid at two different concentrations, what it clearly shows is these parameters have come back towards normal, or it has significantly reduced, indicating that bempedoic acid has some role in the treatment of NASH. Moving to the next slide. Talking about analysis of gene markers in liver samples, if you see here, we have the markers for both inflammation and fibrosis, which clearly show elevation in the diseased animals and some reduction with the treatment in these particular markers, indicating the possible beneficial effects coming from the test drug or the bempedoic acid, maybe through these mechanisms. Moving to the next slide. Talking about the histopathology in this particular model, so what you see in this case is the HND staining. If you see in the top center panel, what you see here, the animals which are diseased animals show clearly the steatosis. And if you see the top right panel, where you clearly see the drug-treated animals showing reduced steatosis, the same with another high dose, you see the animals in the panel in the bottom left show again the reduction in the steatosis scores. In the bottom center panel, you see the animals having the ballooning, which either was shown very less or no ballooning seen with the test treatment. And if you also see the NASH scores, which is one of the key scores which people adopt in the scoring of the NASH, again, you see clearly there is elevation of NASH scores in the diseased animals. And upon treatment with the test drug, you see there's a clear reduction in these scores, indicating there is a clear beneficial effect with these particular drug in this animal model. Move to the next slide. So now talking about CDA-HFD diet-induced NASH model in mouse. This is a model of diet again, and there is a modulation of the choline deficiency in this, the L-amino acid diet. So what you see here is clearly these animals will have a lot of higher fibrosis as compared to other two models which I just spoke. So what you see here is these animals are subjected for this particular diet for about 7 weeks. At that point in time, you randomize these animals on based on body weight, ALT and AST, and then the animals will be subjected for treatment for the next several weeks. In this case, we have tested telmisartan. And at the end of the study, you sacrifice these animals and collect liver and subject it for histopathology and various markers analysis and the blood parameters. Moving to the next slide. Again, talking about liver health, in this case, what you see here is the yellow bars which represent the diseased animal in this case. So in this case, what you see here is the liver triglyceride, cholesterol, or the relative liver weight are all elevated in this particular condition. And then upon treatment with the test drug, the telmisartan in this case, these show significant improvement, indicating this molecule has a beneficial effect in this particular model. Moving to the next slide. Talking about liver function tests and gene markers, again, in the diseased animals, you see clearly elevation of plasma ALT, AST in this model, and liver col1 expression is also elevated, and some of these parameters have reduced with telmisartan, indicating again there is beneficial effect with this particular drug. On the next slide. So now I just want to showcase some of the posters and publications which we have made our drug discovery or the animal model development efforts into publications, so that people from various backgrounds can visit these publications and get to know about this, and also understand our capabilities and reach out to us wherever you need. We can collaborate and help you on your drug discovery projects. Moving to the next slide. So this is about the infrastructure we have to support the NASH pharmacology, as I just enumerated. To enable all that what we have presented, we have an expert drug discovery pharmacology team who have several hundreds of combined research experience, who have supported drug discovery programs for the various biotechs, mid pharma and the large pharma over the years. And we have successfully taken molecules in the metabolic space into preclinical candidates, and later the claims have taken these programs into phase 1 and phase 2 trials. So significant experience in the metabolic space, and NASH is available within the scientists at Jubilant. We also have the state-of-the-art facility which will support this particular pharmacology or the animal models. And we have access to different animal models around the globe, and we are a LAC accredited animal facility, and we are very flexible in our approach to collaborate with our potential customers. So we look forward for our interactions, and thanks for your taking time, and I'll be happy if you have any specific questions we can answer. Thank you.
Thank you, Sanjay, for taking us through this interesting topic on NASH and animal models in NASH. As we move on to the Q&A section, we will be launching another poll on your screen. Please make time to respond with your queries, with your choices. And for those of you who would want to ask any questions to us, please type them in now in the Q&A section. We have already received some questions, so we'll begin taking the Q&A momentarily. Thank you. So moving on to the Q&A section, Sanjay, we have a handful of questions. We'll try and handle a couple of them now, and then the rest of those who are not answered live during the webinar, please rest assured we will get back to you by email. So the first question is asking about non-invasive test in NASH. Can you elaborate a bit more on those lines?
Yeah, it's a very good question. Thanks for whoever they have asked this question. And as I said, non-invasive tests in NASH is very essential. It comes in two things. One, when we talk about invasive, it is mainly the liver histology. So as you know, that liver histology is a very painful process and very expensive, and you need expertise and facilities where people should be very carefully take these procedures. When we talk about non-invasive procedures, what it mainly means is you either take about samples from the blood and you investigate these with multiple techniques. So, and also there are something called as imaging techniques. As I said, there are more than 40, 50 markers or the imaging techniques which are available to drug discovery community in these days. But what is important in the last several years is that people have figured out which of these are more translational or it mimics the most part of the NASH progression and the various stages of the NASH. So people in most recently, they have identified markers, and also if you see in the oncology space, the liquid biopsy is taking a good shape, and similar approaches are taken in the NASH space, though it is in a very nascent time. But what you can expect in the coming years is a lot of investment both in the academic community and the drug discovery and the development and diagnostic community are working towards identifying non-invasive techniques where staging of the disease and prognosis of the disease can be made using blood samples, which are very easy to draw, and it will be very quick to give the results for the community.
Thank you. Thanks, Sanjay. So the next question is from Heather. And the question goes as, can you elaborate on Jubilant's capabilities relating to lipid and inflammatory support for in vivo models? And also, can you evaluate VLDL?
Yeah, yes, we have the lipid panel, and basically we have a lot of, so starting from the HDL, LDL, we also have, we have a corrected factor for VLDL, but we can work out and try to get these things. And as you know, we are a very flexible company. And if anything is not validated within our present capabilities, we are very open to quickly enable these things for our customers. And we'll engage with you and support these things.
Thank you. So the next question is asking about what is the support that Jubilant can extend around additional markers in an efficacy study?
Yeah, this is a very important and very interesting question. As you know, today I was just elaborating about the markers which are available for the NASH space. So what our expertise here is about various markers of fibrosis, inflammation, and the metabolic markers we have. As you know, there are several markers. It is limitless in various markers. But what we have the state-of-the-art multiplexing techniques like MSD, where it can accommodate several inflammation and fibrosis markers. We also have something called FACS, which is again can provide us the markers of both inflammation and the immune side of the things. And we have the several other techniques which we can combine, or we can select one of them. And other things what we can do is we can also ship our samples for the customers or to the third-party side where there are specific things which can be enabled. So we'll be very flexible in accommodating the request of what we can enable within our expertise. And we can talk to you and ship the samples to your respective claims, to the claims or the third-party side, where some specific things which is of most value can be done outside. Thank you.
Thanks, Sanjay. Another question comes from Sotirius. I'm not sure if we can answer it extempore, but if not, we'll come back with a more elaborate answer by email. So how do you think that bemboic acid works? And is there human data that confirms transferability of this model?
It is a very nice question, and I think when we just wanted to undertake the validation of this particular model, we went through various drugs and things. So what you observe in this particular thing is this is acetylation, ACYL pathway, which is it intervenes, and then it works through this pathway, where it is known that intervening with this pathway, you have beneficial effects. And bempedoic acid has partly responsible to inhibit this pathway. So we feel that whatever the effect of what we have seen in this particular model is coming from there, along with other metabolic effects it has. Thank you.
Thanks, Sanjay. We have several questions, but in the interest of time, I'm going to take one last question. And the question is around, does Jubilant Biosys consider developing new animal models?
Yeah, in the past, it is a very good question. And yes, we have done this for our selected claims, depending upon the scope of the work. And we have closely worked with our collaborators, and we have developed and validated. And it is regularly used across various therapeutic areas and also in metabolic diseases. We'll be open to discuss and understand the feasibilities, and we can discuss more, and if possible, we can develop that. Yeah.
Thanks, Sanjay. For paucity of time, we will not take any further questions. For those who have not heard the responses, please rest assured you can expect to hear from our team. For any one of you who wants to inquire about further about Jubilant Biosys' offering in the small molecule drug discovery space, please feel free to reach out to us at bd@jubilantbiosys.com. One of our BD colleagues will be happy to get in touch with you and provide further information. So with that, we'll close this webinar. I'll want to thank every one of you to take time and attend this webinar. For those in the stateside, I'll wish you a happy Halloween this weekend. And for those of you who are in Asia or India, I wish you a happy Diwali, and may this festival season bring cheer and prosperity to all of you. With that,